All right. Hi, everybody, on this Thursday morning. It's my pleasure to introduce Zifeng Wang, who holds a PhD from University of Illinois Urbana-Champaign and is currently the co-founder and head of AI at Keiji AI. And Zifeng is going to be talking to us about use of AI for clinical development. Go for it.
Okay,
And sorry,
thanks.
one more thing. Sorry, Zifeng asked that if there are questions during the talk, you can put them in the chat, and I'll make sure to interrupt Zifeng to let him know that there's a question in case he misses it.
Cool. Yeah, thanks, Noemi, for introduction, be the moderator of this session. And good morning, everyone. My name is Zifeng Wang. I'm very grateful for the opportunity here to present at Columbia DBMI about my research in empowering clinical development with large language models and AI agents. Okay, so let me begin with the broader context of my research. Clinical development is a critical stage along the drug discovery and development process. And we can see from this funnel, in the early stages, we usually identify drug targets and try to design and optimize our compound molecules and select the ones, go to the preclinical research and collect the safety profiles with animal model testing. And after that, we go to the clinical development process. We'll have to initiate an investigation of new drug application, conduct clinical trials, collect the clinical trial results, then submit the new drug applications. So far, this process has been extremely costly and risky. On average, there are some numbers people have collected and analyzed. It's like on average, now it takes $1 billion US overall bring a new drug candidate to get approved by FDA and commercialized. And this process, meanwhile, is also very time consuming. It takes like on average a decade to make this happen. But what makes it worse is the probability of failure is also very high. 90% of drugs, even if you have passed through this 5 to 6 years of drug discovery, may still fail in the clinical stage. So overall, together, our motivation here is to explore whether we can leverage AI to streamline and optimize the clinical development. And we can consider the challenges along the 5 key steps along the clinical trials, like in the planning, design, execution, result analysis, and final FDA submission. To illustrate this idea, I put this overview of my research along these 5 key steps. So in the planning stages, we have developed methods to extract and synthesize evidence from scientific literature and real-world data to support decision-making in clinical trial planning. In the design stage, we have started leveraging historical randomized control trials to make clinical trial design suggestion and optimization. During the trial execution stage, we focus on improving patient recruitment by making AI to do patient-to-trial matching, or on the opposite side, from trial to patients. In the analysis stage, we have developed predictive and generative models to simulate patient outcomes or to simulate the full patient trajectories. In the submission stage, we investigated into the compliance and the factual accuracy in the clinical trial documents made by AI, and how they can be mitigated through a specialized AI system and LLM as a judge system models. So let's begin from the first stage. And this is about the clinical development planning. And we have known that this process is heavily dependent on the evidence collection and analysis. For example, if we start a new clinical pipeline, we usually start from analyzing if there is any gap in the medical practice, which we can call unmet medical needs. So this can come from analysis of real-world data sets and treatment guidelines, like clinical guidelines and so on. Once a potential gap or like indication is targeted, we can look into a library of drug candidates and evaluate which molecules may be helpful or promising to address this gap. And this requires us to examine the evidence about the drug's mechanism of action, biological rationale, and preclinical studies. Furthermore, we need to understand the competing landscape of this new drug along these therapeutic areas. So this will include the analysis of competing drugs and competing ongoing trials, as well as understanding the market size, as well as the regulatory issues and guidelines. So across all these steps, we can find they are all about where to find the data sources, how to extract and synthesize evidence from these heterogeneous data sources, and help us to make effective clinical development planning. So in our work, we primarily focus on two major data sources so far. The one thing is the literature and knowledge bases. The other is the real-world patient data. So to tackle this problem, we can decompose the evidence extraction and synthesis process into a pipeline with these three key steps, right? We have the raw data, and we find the data representation. Then we extract the structured data, then leverage the data to do the evidence synthesis. So let's begin with the literature and knowledge bases. A common expert workflow for this task is to do systematic literature review of historical randomized controlled trials and their corresponding clinical studies. So this is an example of a Prisma framework, which we start from identifying the studies from different literature databases, then define the eligibility criteria to do study screening, and then involve the studies for data extraction and quantitative evidence synthesis. If we look closely at this process, we find all of these steps can be decomposed and be handled by specialized LLMs or AI agents, which is well aligned with the design pattern of using AI or LLMs to handle this research type of challenges. So with this idea, we decompose this workflow into several steps. Right? First, we have a research question, which may be formulated in a pico elements. Then we can have AI help us do the study search and screening the studies, extract the data, and perform evidence synthesis. So this could be handled by an agentic workflow, while you have specialized AI agents with different expertise that just focused on from research question to find the studies, or from the candidate studies to find a subset by which we should include, and so on. And meanwhile, this design has made it possible for a human expert to monitor, right, to define the inputs at each step and monitor the outputs, then correct the AI agent's errors before you proceed to the next step. Because this kind of agentic workflow chain can accumulate errors if we don't take interventions in the early steps. So here is a demo video we have developed to enable this agentic workflow working with experts. So we can start from defining a pico elements type of research questions. Then the first agent will help us generate a set of keywords covering the conditions and the interventions, then provide a comprehensive search of the clinical trial studies. After that, the second agent will help us formulate the eligibility criteria of the studies, then estimate the criteria of each studies according to these 5 defined criteria. And then this can help us to quickly screen and pick the top ones. At the last step, we have AI agent that help us extract the structured data at scale from all those publications. And at each data field extracted, you can see the sources and to validate and correct potential hallucinations or missing elements in this extraction. So you could say in this way, we can unlock the power of AI agents in this review process, but meanwhile have human in the loop to actively take interventions and monitor the whole process. Sorry. So we have made the system work with a lot of clinicians from different medical institutes and have some results testing how AI agent and human collaboration can reduce the time and improve the performance for the study screening and data extraction process compared to the fully manual process. So in our previous work, we have formulated the review task into 4 steps and then implement an agentic workflow. But that has been based on a generic LLMs with in-context learning, like specialized prompts for different steps. The next question then we asked was, instead of just using a generic model, can we just develop specialized model optimized for these steps, right? We do find some performance gap there because of the limited capabilities of generative model. And with this idea, we developed this foundation model called LEAS. And that starts from collecting the data sources from like publication data. We have 20,000 review papers collected, and with 400,000 studies involved in these review papers. So we can leverage those links and create a bunch of instruction datasets covering the study search, screening, and data extraction problems. So using this instruction data, we conduct a supervised fine-tuning of a 7 billion model. And along these dimensions, we find that even this data are public available. And probably GPT-4o or this generative model has ever seen this training in their training corpus. But still, if we can organize this data in a format that well aligned with our research tasks, the fine-tuned model can still outperform the generic model on those tasks and with all-round performance improvement, even with much smaller model sizes. Now let's move on to the other data sources, the real-world patient data. And in that dimension, we have mostly focused on how to make a representation learning on the data and support extraction from that data. Our first work along this direction is MACLIP, where we have investigating to conduct contrastive learning between medical images and text, two very important modalities in real-world data. And the challenge here we find is, first, the paired medical image and clinical notes are usually limited. And the other problem is, even we have paired data, and if we use the traditional contrast learning approach, we will treat the other unpaired, right? If this is an anchor image, the other clinical notes we'll consider is a negative example for this anchor. But if we have like this medical image and text data, you may find sometimes the other clinical notes may still describe some clinical concepts that appear in this anchor image. So that can introduce a lot of false negative examples in your contrast learning and undermine the performance. So to mitigate this problem, we have tried to create another supervision based on the clinical concepts either contained in the medical image, like those labels of the medical images, or the key entities in these clinical notes. And then we have them compared along this same semantic space. Basically, we have this clinical concept. They are like a set of keywords. Then we can calculate the distance of arbitrary medical images and clinical notes and compute this semantic matrix as a way to train our like medical image encoders and text encoders through this contrast learning approach. So as a result, we have found this model has made a significant improvement on the data efficiency, which means in the same amount of data, it can get better performances than the previous methods. And with more data, it can scale better with this new like supervision approach. So beyond the images and text, we might also explore other modalities in this domain, like drugs, proteins, right? Like gene ontologies, disease. And this setting is even more challenging because considering so many modalities, we may even don't have any paired data that contains all these modalities together. So considering this challenge, we have to leverage the other knowledge sources to help us build the contrast learning. We have leveraged biomedical knowledge graphs because they contain a lot of relations between these modalities. Like you have like a protein associated with a disease, so they connect different modalities, and in this way we can extract these triplets from this knowledge graph, and then consider we have a single modality protein embedding model, and we have another like disease embedding model. Then we can project this protein embedding to the disease embedding space using our BioBridge module. And then this H hat tail will have to push to the closer to this H1 tail, which is our target embedding space. So this helps us build this like contrast learning to push them closer. And meanwhile, we can sample inactive disease entities which do not have associated with relationship with a protein, and then push them away. So this formulates the supervision for our BioBridge module. So in this process, we'll keep the protein, the single modality protein foundation model frozen, but just fine-tune this bridge module. As a result, this BioBridge module can serve as a cross-modality retrieval system. So now use logic graph as a supervision. We no longer need a logic graph to do the retrieval. So it can work like if we have a protein sequence as an input, we can encode this with a protein embedding model and project this embeddings to the disease space and then retrieve the most similar diseases. And using this all as a context plus the input prompts, we can use this as a retrieval augmented generation approach from another LLMs to give an answer for our questions. And similar for another like scenario, we have a target conditions, and then we can project this condition to the protein space and retrieve the relevant protein as a potential targets. And from LLMs with both the protein targets and input condition and try to generate some molecules. So in this way, BioBridge has become a unified interface that connects diverse modalities and as a great plugin for an LLM to answer this type of question, taking multimodality data. So here we have covered the planning. In the planning stage, we have mostly working on the literature and real-world data and on data representation, learning, extraction, and synthesis from along different dimensions. And then we can go to the next step. How do we leverage LLMs or AI to support clinical trial design? So when we talk about clinical trial design, we are mainly referring to the protocol of clinical trials. So that is a really comprehensive document, contains a lot of different aspects of a trial, like what's the objectives, right? What's the context, and what's the eligibility criteria, the schedule, and so on. So supporting this trial design is a very challenging task. In practice, even the human researchers will try to leverage some reference historical trials, right, as an input and maybe as the baseline, and then iterate over that to design the new clinical trial. So before we develop model to do that, to generate criteria design, we want to start with how to supporting an effective search of clinical trials, so we can provide a reference. So this motivates our work trial to VAC. So in this project, we leverage a large-scale historical randomized control trials design from clinicaltrials.gov, but we connect them by taking the shared medical entities between the trials and use these shared entities, make some replacement and perturbation. So this can help us build very sufficient number of contrasted learning positive negative pairs to train our embedding model. And that embedding model has become, if we make a t-SNE plot of the trial level embeddings from our model, we can find they have really captured really meaningful like semantics along of those trials. And this embedding model has give us a better trial similarity search performance compared to the generic text embedding, like those sentence embedding models in this type of task. Building based on trial to VAC, we can further explore leveraging AI, like large language model, to generate clinical trial design. So we start with this eligibility criteria generation. Eligibility criteria is basically a set of statements, right? You define the inclusion and exclusion criteria about the target population you want to recruit for your clinical trials. And the challenge there is, at the time of when we were doing this research, we only have GPT-2 level of language models, like small language models. So for those models, it's really challenging to generate this kind of criteria, this long criteria, just taking a input trial setup. And so to make this work, we add this trial to VAC as a retrieval augmented generation system. And we make sure our GPT-2 model can learn to do this retrieval augmented generation in both the pre-training and this fine-tuning step. And we introduce a continuous prefix tuning approach. And so this model can take the trial setup as prefix and then use trial to VAC to include the similar reference trials criteria in the context, then generate or suggesting the criteria for this input target trial. So this setup has made, at that time, an auto trial model based on a small language model and to enable us to generate meaningful eligibility criteria design that the other models may generate very off. based on our evaluation. So beyond just generating trial design, we also try to predicting the clinical trial outcomes. And so this setup is more like if you have an input trial setup, including the target drugs, the target disease, and what type of population, and the other design elements as input, can we estimate the probability of this trial if the proposed new therapy will get FDA approval at last? So in Spot, we have incorporated two key insights that beyond just takes this individual trial setup as input. At first, we see that the temporal patterns of this trial really matters about the final success of this trial, because if we take a clinical pipeline as an input, we may find it may have previous trials, maybe another phase 1 trial, or you may have multiple parallel phase 2 trials, and their results and their patterns has great impact on the success or on the performance in this clinical trial, in this current clinical trial. And another aspect is the competing landscape, because you may meanwhile have a lot of other sponsors who are working in the same indication area, but maybe have different molecules with different mechanism of actions, and they have different working pattern, or their progress may vary. So this has a great impact on the final outcomes of these current trials. So consider these two insights. We want to resolve that with machine learning models. So that introduces the core ideas of Spot, right? We use trial to VAC as a way to encode our clinical trial embeddings and then cluster them to find the competing landscape of the clinical trials. And meanwhile, we can group this clinical trial by their time steps to get this sequential pattern, make a recurrent neural network to model the sequential pattern of these clinical trials and make the final trial outcomes predictions. So these two approaches have allows our model to achieve significant better performance than predicting trial outcomes than these baselines, which do not consider these temporal patterns and competing landscape. More recently, we tried to build a foundation model to cover the previous trials, previous tasks we have talked about, like trial search design and outcome prediction.
Sorry, I'm interrupting you. There's a question in the chat. Adi, do you want to just say it?
So I noticed in your Spot work that there's a emphasis on accounting for different phases when it comes to outcome prediction and modeling. For auto trial, was there any consideration of kind of design that differences depending on the phase of the trial at all, or was it strictly from a condition, target, and intervention standpoint?
Yeah, thanks for the question. So we do not discriminate trial design by phases, but we do discriminate. I mean, we do only involve the interventional trials, not the observational ones. And the inputs may include the basics, setup, the clinical trials title, the target diseases, treatments, and those kind of those information covered in the synopsis.
Okay, thank you so much. That clarifies things.
Yeah, thank you. Okay, so let me continue. In this trial panorama, we try to build the foundation models to cover the previous this trial search and design and outcome prediction task. And this can be achieved only by a large scale data foundations. So we collect a lot of clinical trial design information from global trial registries like clinicaltrials.gov, but other countries, regions, and as well as some publication of clinical studies. And then we aggregate those data to formulate a knowledge graph of these clinical trials, because we can find the links between a publication and clinical trials, or links between a review paper and included studies. So this can become a graph of those clinical trials. And also we standardize some components of this clinical trial and formulate relational databases to contain all of this clinical trial information and connect them together. So this is really a rich data set covering over 1 million clinical trials and from very large sources. And with this data foundation, it's become more straightforward. We just build instruction data sets by taking some trial setups as input and extract a target setup, let's say the sample size as a target. Then this become a sample size estimation instruction data. And so to show the utility of this data set building way, we have create a lot of like instruction tasks such as like study search screening, like we have covered in literature review, as well as some design generation tasks like sample size, criteria, endpoint, arm, and also estimate the trial outcomes. So of course, with this large data, we can definitely train a very good models. Like we actually proved that in this setup, we have a Q13 like 8 billion model using supervised fine tuning and plus some reinforcement learning with verifiable reward approach. And this model has outperformed generic models such as GPT-4o and also like more generic open source model like 70 billion llama along these dimensions. So this is just another demonstration of how even a public available data in a good organization and instruction data building can give you better vertical models. Even this data are available in the training corpus of a generic counterparts. So far, we've discussed the trial design, like how on generating trial design and optimizing or like predicting trial outcomes. Now let's move to the execution stage, which we focus on patient recruitment. So patient recruitment is definitely a very critical, a well-known bottleneck in clinical trials because every stage is you have recruit some number of human participants to assess your drugs, right? But in phase one, you may need tens of them, but in phase three, you really need hundreds, thousands of them. So that's really a bottleneck that caused a lot of trials delayed, or even caused some trials terminated because just cannot find enough number of patients. And meanwhile, this is also major contributor to the overall cost of a clinical trials. So that's has just given a lot of motivation on like, can we use AI to streamline this process, right? Then, so based on this motivation, we developed Trial GPT. So this is an agentic workflow that can match patients to recruiting clinical trials. So here's a demo. We just basically show how this input works. Right? We take an input clinical notes, just free text of the patient description, and the Trial GPT model can help us search the clinical trials and give you a criterion level assessment again, like comparing this input patient notes against the eligibility criteria of each trial. And after that, it will also give you an aggregated like score of the eligibility of this patient to each of this trial. And this provide a really transparent way for you to invest to inspect the agent's outputs, and you can select. So this also streamline the process for if there is a PI or a clinician who recommend a patient to possible trials, right? You can give you a very short list of candidate trials and just improve the efficiency. So we also conduct a user study with NIH investigators who are running 6 clinical trials, and we compare Trial GPT versus manual efforts in matching their patient inquiry to their clinical trials, and this has shown Trial GPT can give you 40% time saving in this like patient to trial matching process.
Zifeng, can I ask a question on this? How do
Sure.
you, the input sounds like a summary of a patient record. Like, do you expect something of that length, or is the system able to work with the real world data where there would be a very long context to the patient record?
Yeah, so far in this demo, we only consider this short informal patient inquiry. So we have not really tested it with a more realistic, a very long context, even a very long context, even multimodal data. But that's definitely, I mean, there's a need for that extension.
Thank you.
Yeah, thank you. Yeah, so in Trial GPT, I think the task is like from patient to trials, right? So that's a very useful application for patient referral, patient pre-screening. But there's the opposite side, right? If we have a new trial, right? You want to assess the quality or you want to assess the feasibility of this trial. Then you may want to start from this target trial and go to the EHR systems to find the target cohort, then estimate or like analyze the characteristics of these populations. And also, if you are actively recruiting patients, you want to go to this different EHR system and find where are your eligible patients, right? Then this can help you do site selection and streamline your participant recruitment process. So this is another problem setup. And you could imagine that if you have a EHR system, the patient records sit in a relational databases, right? Then this can definitely benefit from if you have a data agent that can help you generate search queries and do some programming to help you do cohort extraction and analysis. So here's a demonstration of how data agent can handle that. So we can start from a input trials eligibility criteria. And the database here is a real large scale clinical data plus genomics data. And these data agents will try to first understand your criteria and get access to the dataset schema, then provide you a selection logic, which basically describes the data sources, what the criteria are applicable to this database and what are not applicable because of missing data, and so on. So this will provide a way for the users to inspect the selection process. And then the agent will try to generate programs and queries to show you the impact of each criterion and the population characteristics, such as the most frequent genomic alterations found in the populations and so on. So after this process, also agent provides you a flowchart of the patient screening process. And we can also inspect the code, like either C code, like those structured query generated, and also the other analysis code generated along this process. The system can also take your any analysis type of questions as a follow-up, right? Here I ask a question like, can you do a funnel analysis of the cohort so I can take a deeper look into the each criterion, what's the impact, right? Is there any space for optimizing the criterion to improve the feasibility and so on? So this is a data agent can help you do cohort extraction and potentially allow you to optimize your criteria and streamline your participant recruitment. Okay, so let's now go to the fourth step on the clinical trial result analysis using AI. So here our research focuses on building patient digital twins using generative or predictive modeling. So traditionally, if you have a new trial, right, usually you need or a most classic setup is you have to balance your treatment and control arms. Right? You have 4 patients in treatment, 4 patients in control. But if you have a generative model, let's say a simulator that learns from a really large scale real world data and can predict very accurate patient outcomes, patient response. So that model can be useful to create a synthetic control arm. Let's say maybe not the only way, but there is a possible way that you take the treatment arm patients and predict the counterfactual situation while the patient were allocated in the control arm, and that can help you recruit less patients in your analysis. And so to make it work, the key methodology here we need to tackle is how we do better predictive modeling. And if we want the full patient trajectory, not just a final outcome prediction, you should consider generative modeling on this real world patient data. So the first line of research here is in our previous work is develop models for patient outcome prediction. And the challenge there was when we consider patient outcome prediction as a tabular prediction task, which means if you have a table and a row of this table is a patient and a column of the table is a patient feature. So that means the data collected from different EHR systems or from different sources, they usually have unique schema. And if you want to make tabular prediction on those type of data, you have to using traditional machine learning models, you have to train unique models on them separately. And that has made it very, very hard to train large models on large data sets because of the data sets are so segmented and heterogeneous. So our idea here is if we can have a foundation model that have a like can train on all these heterogeneous tables, and then these models can leverage really large scale data, then it's possible that this model can do zero-shot prediction for new data set. So that new data set can be a clinical trial. That's our motivation and the method we have developed in our series work, the MediTab and TransTab works. So I will just show the results here. We developed a model on a EHR data for mortality prediction, and then we use this model to predict on 7 clinical trials. And we find that when we compare that zero-shot prediction from our models compared to dataset specific XGBoost, which is a very powerful machine learning model on tabular prediction, it has actually win over this dataset specific model on 5 of the clinical trials. Because one reason there is the clinical trial data size is super small, so it's very challenging for machine learning model to be fitted well on this small data set. But the foundation model doing this zero-shot prediction, because it's trained on very large EHR data, it can potentially extrapolate to some unseen like clinical trials. The second line of work I want to introduce here is doing generative modeling of patient trajectories. We call it prompt EHR. And the key idea is if we have a EHR data, can we find a way to just transform that EHR, like this patient records, to a sequence of event tokens? And then in this way, we can use transformer to do like just causal language modeling. And when you simulate patient trajectory, it's just like you provide the patient baseline informations and then simulate the sequence of tokens as a patient events. And to make this prompt tuning, this prompt-based generation possible, we introduce a prefix tuning approach that takes the patient baseline features like their demographics information, for example, as input, and then have this prefix as a inject the prompts embeddings there to enable our model to generate more personalized patient events. So this model can then work like you have a first visit, for example, then have some baseline information, then the model can do next token prediction to complete the next events of this or like next visits events of this patient. All right, then the last stage is like the submission.
Sorry, can I ask some more questions on the generative
Sure.
model?
Sure.
So you said, maybe I misheard, but you said causal LLM. Yeah, you said causal language model. What do you mean by that?
Oh, so this is just a terminology we borrow from language modeling, because like causal language modeling means you have the like several words in the sentence then predict the next word, right? That's all it does. And here we want to do is you have the first visit of the patient, then you want to predict the next visit, right? Then the next visit is consists of a couple of events like diagnosis code or like lab test code, example. So we call it causal language modeling, but it's not necessarily language modeling. It's like
Right, it's an
patient.
autoregressive model, but
Yes.
autoregressive, like there's no real, like it's a language model. It's not a causal language model in that
Yeah,
case, no?
it's a language model. Yeah, it's a like
Okay.
next token prediction. We also do next
Okay.
token prediction, but token is medical events here. Yeah.
Right. Okay.
Mm.
And can I ask you a question about the evaluation? So typically in this kind of foundation model or generative models, the longer the trajectory or the sequence that you're trying to generate, the more chances for, getting it wrong, basically. There's also a question here of like, what does it mean to get it wrong or well when it comes to a patient trajectory? Could you talk more about these two things?
Sure. So here we in the original public paper, we investigated this as a synthetic patient data generation task. So that means we assess the quality of the synthetic patient data by computing the fidelity of the data. So that fidelity is like we can estimate the marginal distribution along some dimensions and compare that against the real patient data. And also there are a couple of fidelity evaluation we have made there.
But in the case, in the case of language, but in the case of an EHR or a patient trajectory, it might not matter if you get the glucose code wrong, but it might matter if you get the wrong disease or something like that. When you compute the fidelity, are you taking any of that into account?
Yeah, so one evaluation we did is we borrowed the conception of perplexity scores from NLP. So here we can use the because this language model is making probability prediction for each token on each position, right? Then we can use that probability um, of the right token. I mean because there is a ground truth records there. Then we can calculate that probability of our model
Yeah, and
of
I
this
guess
right token.
I'm saying that the perplexity is taking every token as ground truth. And
Mhm.
if we think about generating a patient trajectory, I might not care so much that I forgot to generate or I generated the wrong lab, or at least maybe it's less of an issue that if I generate the wrong disease name or the wrong disease code. And so perplexity scores don't pay attention to that. They just say every token is ground truth. Did I get it right? Or did I get the sequence generated right? Have you thought at all about like what it means that what you're generating is not a sequence of codes where every code is as important as the other?
Yeah, so in this paper, we do not currently consider the difference across difference between the event types. We just treat all of them as a single token and just calculate the average perplexities. Yeah, but yeah, that's a good point, and I think yeah, maybe some events are more important to capture, and we haven't taken that into consideration so far. Yeah.
Right. Thank you.
Yeah, thanks for your question. And yeah, so I will go to the next stage about the regulatory submission, right? You have a lot of documents you want to prepare, and definitely people are trying to use the large language model to generate those documents. But there indeed introduce several risks that should be carefully addressed in this highly regulated space, right? The first risk is the hallucinations. So we need to check the factual accuracy and introduce a judge model to assess the element level accuracy, like statement level accuracy to calculate the factualness scores. And the other unique challenge there is the document should be compliant, right? You have to follow some regulatory guidelines. And that's another compliance check we introduced there for the document drafting process. So with these two judge model as a verifier in these systems, we can introduce a more robust agentic workflow to generate clinical trial documents, and with this judge model to monitor the performance and push the agent to do more refinement. And we do find this iterative process has made our agent workflow to achieve better performances than the ones without this judge model as a additional verification. Okay, so far we have walked through all the 5 stages, and now I want to briefly discuss the remaining steps or future work along this direction. And so first, let's review the fundamental challenges in clinical development, right? We know that it's very costly and risky and takes a long time. So the first principle is we need to find a way to improve the probability of success of our drug, like clinical pipelines. And meanwhile, is we should try to control the risk along different dimensions and also control the operational and the opportunity cost along this process. So with these principles in mind, we can imagine what type of AI methods or workflows will be helpful to improve along these 3 dimensions. So specifically, I would like to share three potential future projects that can be interesting to explore. The first one is multi-agent system for clinical trial intelligence and support plan and design of clinical trials. And second one is extend our data agents to be more professional and effective to work with real-world data. And the third one is on the real world and on the patient digital twins perspective, can we train a really large, powerful foundation models that can extrapolate very effectively to clinical trials and generate trial-specific digital twins. So in the first future work, our focus is on the clinical pipeline development or process. And this clinical pipeline development is really about how to define your drug candidates in terms of the target population and indications. And you can see even the largest pharma, they cannot maintain a very large number of pipelines. Like AstraZeneca may only like have 200. It's really very big. But the other ones, which are also even bigger than AstraZeneca, may only have like tens of these drug pipelines. So this just show us the clinical pipeline development decisions are really high stakes and requires a very comprehensive assessment of different dimensions along like and you can find information from literature, from previous trials and so on. So with this challenging mind, I think our idea is, can we build multi-agent system that can mimic the human teams working farmer in terms of the analyzing the risk along the categories, different categories and different data sources. So if we take this clinical risk assessment as an example, one typical outputs of like this of sponsors in this research process is a drug benchmarking table. So their data sources are the literature, historical randomized control trials, some knowledge bases, and they need to collect a lot of this type of information and try to consolidate them into an overview of all the competitors' drugs and also the approved drugs and along different dimensions on the what type of population, right? First line or second line or like this type of information, as well as their primary results, like their primary endpoint results and so on. So this, you could say this process is really about the clinical trial intelligence. And we want to build multi-agent system that can accelerate this process and provide to acquire the evidence and synthesize evidence to support decision making in this clinical pipeline development process. And to achieve that, the key is to effectively or efficiently sift through all different types of data sources. And this definitely is not the challenges that unique in this like this clinical pipeline development. So in fact, we have explored this in our recent works on developing a deep research system that can effectively do wide and deep research on like different knowledge bases and like biomedical knowledge sources and do like cross-source alignment and evidence synthesis. And meanwhile, we also provide evidence map building capabilities of this system. So it will show you what type of data sources it has identified it, and what reasoning paths it has to along this research process. Based on this foundation, our future direction is to extend this multi-agent deep research framework to be more specialized in clinical trial plan and design. So that will involve like in integrating more data sources, not just this literature or knowledge bases, but also like real world data, for example, and find more specialized like orchestrator that can handle the tasks like this competing trial or drug benchmarking task we have introduced in the previous slides. So in the second project is on the data agents for clinical development. And before introducing the how the future perspective, I want to like give a concept about what data agent means, right? We have seen the demo show the data agent generate programs and extract a cohort. And this is that demo is built based on our previous research DS Wizard on streamline the biomedical data analysis. And this is a concrete example on biomarker discovery using genomics data. So a human expert may take like multi-step iterative process on analysis with this genomics data. And the data agents is the one that can help you do autonomous data extraction and analysis. So with this help of data agents, a human expert can just set a target and review the results. And a data agent will automate all these intermediate data analysis steps. One example here is we have extended our data agent for some autonomous biomarker discovery. And the right-hand side is the technical report generated by our data agent last year on taking a cancer genomics data set and try to do a whole bunch of like analysis such as the differential expression analysis, patient subtyping, understanding the mechanism, or like do gene set enrichment. And we find that data agents are like, because LLMs are more powerful, and this data agent become more and more powerful in like this autonomous discovery. But considering extending this agent to real world data or in clinical research, we may consider observational data science as an example here, right? There are definitely some gaps we have identified to be done, to be filled, to make this data agent useful along these several dimensions. The first one is the underlying capabilities of this agent system. It has to work well with the structured data, which may be from different common data models like OMA, FIRE, and some clinical trial CDIX data set as well, and also some unstructured data format like clinical notes, imaging. So that's the essential like format requirement that format exists in real world data. And this data agent definitely should be able to leverage different programming tools like R packages, Python packages. It requires to do like professional observational study. And for this unstructured data, we may consider develop some specialized machine learning AI tools that can be leveraged to extract and do retrieval of this type of unstructured data. And in the learning perspectives, we should consider, can we have our agents not just equipped with a fixed prompt, but by a learning from the past experience, like those published papers, or have human feedback, as well as like if we can develop some research challenges with computable rubrics to drive our agent to do self-learning. So over time, if we have all this training data collected and rubrics defined, we want our agent to be able to like organize and consolidate this experience into some workflows that the agent can reuse in the future. That will really make the agent that can perform expert level like observational research. And finally, we want to assess the real world utilities of these agents. We want to work with like experts in this domain and propose some research question which maybe belong to the type like find the potential indication expansion opportunities of existing drug and so on. So that's the overall system development and how to evaluate this type of data agents. Then lastly, I will just go through the build the foundation models for patient digital twins. And of course, we have talked about patient outcome prediction and some EHR, like do generative modeling on EHRs with prompt EHR work. And in our previous work, we have investigated into this task by introducing a model named Twin. And the challenge there is the clinical trial clinical trial data sets are typically like smaller. And so here we have involved like a phase 1 trial and phase 3 trial as our training and testing data set. And so to handle this issue, usually you cannot use a large scale model to do that because this data is just too small. So we introduce a smaller like machine learning model that do patient digital twin generation by perturbing the real patient records. And that has introduced some good results on the utility of this augmented patient data. We have trained this like prediction models using the data augmented by Twin compared to the model trained on the original trial data. We find that Twin has provided more like utility, which means like the predictive model can become better with this like patient digital twin augmentation. But that's not the whole story. Like if we want to really do patient trajectory simulation before the trial starts, right? Before you really have the before you have the real clinical trial patient data, you still need to borrow the knowledge from the real world data sets. And so make this happen, we want to approach this task along the pre-training and post-training perspectives. So along the pre-training, we should consider how to design the tokenizer for broader and heterogeneous real-world data sets. And we know that in language modeling, we have like word tokenizer, in vision modeling, we have patch tokenizer. So what's the ideal tokenizer for this heterogeneous real-world data set is still, I think it's an open problem. And also the model architecture, you can consider a variation of transformers, just analogous to the other models you can see in the protein modeling and so on. So that's open problems in pre-training paradigm. And also in post-training, if we want to generate trial-specific patient digital twins, you should consider the trial specification, right? So those specifications can be from the clinical trial estimate, define the population conditions, primary endpoints can also come from the schedule of the clinical trials, which define what type of procedures will appear in the clinical trials and in which order they will come in the temporal sequence. And also, you want the clinical trial data to be aligned with the standard data model, like a CDX type of format. And this model should also do post-training and can generate like standardized like clinical trial patient data. So that's the kind of the conclude all the future work I've discussed today. And with all of that, I will thank you very much for your attention and happy to take any question if you have. Yeah.
Thank you so much.
Thank you.
Do we have lots of clapping? Thank you everyone for clapping virtually. Any questions for Zifeng?
Can I ask one clarification question? In your future work, you said in the probability of, I guess, regulatory success as future work. I'm trying to understand from the pharma, like you said, maybe we should have workflows depending on what the expert really needs. What are the bottlenecks that like biostatisticians or regulatory people are actually facing? I mean, the whole talk is great, and I really like it. I just want to understand, what are we augmenting that they don't currently have, or are we just accelerating through the agentic systems what they already do, but we just want to do it at scale so that it will become more efficient? Or there is some gap that maybe they don't have access to, like as much prior knowledge, and we are offering more prior knowledge to aggregate better evidence, or what is that gap that we are trying to fill here?
So thanks for your question. I think for biostatistician, their workflow is on, because they have to build some statistical models on estimating the probabilities. So those models will require a lot of prior knowledge about some key parameters. What's the range or what's the value you should pick for those parameters? So that process can be definitely benefit from if we have an AI agent that access a lot of publications and provide data extraction and provide more solid evidence on this data range and so on. But currently they may just manually pick several papers and just based on their experience set up some value ranges. So that's a way that AI agent can help in just on evidence acquisition and the synthesis. But there are indeed some cases that for not biostatistician but for clinician or like for business analytics, they may do more qualitative assessment of their results. But AI agent may possibly to provide more structured and mathematical as like this quantitative assessment along their dimensions and so on. So that may be make it possible to make this decision-making process more solid, not just like purely based on manual experience.
So what's the kind of quantitative assessment that we're talking about? Is it like we can do the methods at scale, that's why it's more quantitative, or are we providing a different number they wouldn't normally think about? Like, what are we talking about here? Just to ground it a little bit more.
Yeah, so for a clinician, if you ask him or her about what's the existing unmet medical needs in clinical practice, so that clinician may consider his or her past experience and try to, and based on his understanding of the clinical guideline, and try to come up with something. But that can definitely be more streamlined by have a autonomous AI agent try to just analyze the clinical guidelines as well as automate some real world data analysis to ground these decisions on the real evidence you find from the data set. So that's making more quantitative, not just based on the personal experience.
Got it.
Yeah.
Any other question from the crowd? Okay, we're actually out of time. Zifeng, thank you so much. Thank you everyone for joining. See everyone soon.
Thank you.
Thank you. Thanks for listening. Yeah. See you.